Welcome back, everybody. Before we move into this session, which I'm very excited about, talking about immunosenescence, I just wanted to tie it to the first sessions that we heard this morning, which were about COVID-19. And we had government leaders and academic leaders talking about the pandemic. And the question is, why are we talking about COVID-19 so much in a meeting about healthy longevity? And I think the ties are the aging-related diseases are going to become almost like a pandemic in terms of the burdens that are going to be on healthcare systems. And the way we're going to need to handle this sort of pandemic of chronic diseases, we can probably learn lessons from how the COVID-19 pandemic was handled. And I think probably a lot of government and policymakers haven't quite realized the extent of the burden of the chronic diseases that our society is going to face. And I think it's really important for everybody at this conference and in this field to sort of start developing policies and science that are going to enable us to tackle this future sort of crisis for our healthcare system. And I think another important part of the COVID-19 pandemic is who dies from COVID-19. It's people with chronic diseases or people who are older. And this highlights dysfunction of the aging immune system and raises the possibility of maybe a new way to deal with the current pandemic and future pandemics that were discussed this morning is to boost the resilience of this aging immune system. And we have a panel of experts that we're really excited to hear about today who are doing research on what is wrong with the aging immune system and what are some ways that we might be able to boost the function of the aging immune system is a completely new way to deal with future pandemics. And so with that, I'll turn it to Georgina.
So hello, everybody. Our first speaker today, it's going to be Laura Haynes, a professor of immunology at the University of Connecticut and an expert on aging of the immune system. So Laura.
Thank you. And I'm so glad to be here. And I'm just going to give a little overview. And I really appreciate the whole focus on COVID. We're going to, for what I'm going to talk about today is another viral infection, influenza, that we work with in both our human and mouse studies. But and flu has been around a long time, way before COVID, and we've been dealing with this with older adults for a long while. And we know from work that's done by a lot of really outstanding scientists in the field, but mostly things that were pioneered by Janet McElhaney, my longtime colleague who passed away last fall. We know that when someone gets a flu infection, and just want to, okay, oops, sorry. When someone gets a flu infection, if they're a young adult and they're hospitalized, they recover fairly quickly. They have a rapid antiviral immune response. They clear the virus quickly. They resolve systemic inflammation fairly quickly. They recover from any skeletal muscle deficits that they might have during the infection, and they return to normal levels of activity. Whereas for older adults, what we see is that there's poor resilience. There's a poor antiviral immune response. There's slower viral clearance. There's slower resolution of inflammation, both systemic and local. There's slower recovery of skeletal muscles, and that can lead to post-infection disability, which is what we see a lot. Older adults, after they leave the hospital with the flu, they need a cane or a walker to help them get around. So we've been looking at what's going on using a mouse model, and it's very similar to what happens in a human. We have young and aged mice. We give them a sublethal dose of flu, and we monitor what goes on. So with aging, we see that there's significant weight loss, and it never actually returns to baseline post-infection, while young mice respond and recover very quickly. We know that there's slower clearance of the virus in the mice, there's lingering inflammatory cytokines in the lungs, and there's less specific antibody, and these antibodies that are produced have reduced neutralizing activity. And then we also see more physical impairment and induction of muscle degrading genes. And just one graph to show you about antibody levels. So this is total flu-specific IgG, and by 3 weeks post-infection, it's really high in the young and really low in the aged, while with the neutralizing activity, again, great in the young, really poor in the aged. So we began to look at germinal center B cells, since that's the source of the highly neutralizing antibodies. And what we found is that looking at influenza nucleoprotein specific germinal center B cells in old mice, we see, in young mice, we get a nice germinal center response. In old mice, it's very poor. And that's really shown here. You can see the green is the GL7, which is a germinal center marker, very nice in the young, very poor in the aged. And if you correlate the percent of starting weight and the number of germinal centers in a half a spleen, what you'll see is that in young mice that don't lose much weight during infection, we get a lot of germinal centers formed. And those aged mice that lose more weight have fewer germinal centers. So there's a really direct correlation with that. And what we thought here was that this could indicate that if we improve the germinal center generation, maybe we could improve the overall immune response to flu infection and improve resilience. So one last experiment from this paper showed that, again, these 2 points, young and aged here, are this 14-day time point. And if you add back young CD4 T cells to an old mouse, you can really improve the number of germinal centers that are formed. And so maybe it is all about the T cells. So we've sort of been trying to focus on what's going on with T cells during aging. And before we started working with flu, we had done a lot of work looking at what happens to T cells with age. And we know that if we take younger aged CD4 T cells, put them into young hosts, even so we have aged T cells in young hosts, we still see reduced germinal center B cells, reduced serum IgG, and reduced affinity maturation. And conversely, if we take young T cells and transfer them into younger aged hosts, we can also see reduced T follicular helper differentiation of the donor cells and reduced germinal center formation. So there are both age-related intrinsic and extrinsic factors that are going to influence a CD4 T cell function. So going back to what's going on with the T cells, and this is from our paper this year, we know that if we look at the flu-specific cells in the lungs, we know that the biggest difference between young and aged in our model is that there's way more FOXP3 expressing CD4 T cells in the lungs in aged compared to young. And so we tried to think of ways to alter this to see if we can improve what's happening in the aged animals. And one way we did that was to just give a vaccination with flu NP prior to infection. And here what we see is that this is unvaxxed aged mice, and the black circles are the vaccinated aged mice. You can see that we reduced the percentage of FOXP3 expressing Tregs to levels that we now see with the young mice. Other things that we've done, we noticed that there were high levels of TGF-beta in the lavage from the lungs. So we blocked anti-TGF-beta, and again, we can reduce the percentage of FOXP3 expressing Tregs. And finally, since TGF-beta can also be secreted by senescent cells be a component of the SASP, we looked at whether we could reduced the level of Tregs using a senolytic, and here we're using D plus Q, disitamab and quercetin, and we reduced the levels of Tregs in our aged lungs, and we also reduced the TGF-beta. So right now what we're doing is looking to see if this reduction in Tregs has an impact on the overall response to flu. And I'm guessing that it might, based on what Paul's work. So just to summarize, what we know is that we have both age-related changes that are endogenous to the T cells, but also the aged environment has direct impacts on T cells. That all leads to poor T cell health when we're looking to generate high affinity antibodies, whether it be in response to a vaccination or an infection. When we have a reduction in the ability to generate these high affinity neutralizing antibodies, this leads to slower viral clearance, prolonged illness, lingering inflammation. It also can lead to negative effects on skeletal muscle, including induction of muscle degrading genes, induction of T cell chemokines, recruitment of T cells into muscle, and that's going to result in prolonged illnesses, more physical disability, slower recovery, and that's a part of our decreased resilience that we see with age. So, what we've been trying to do is target CD4 T cells to improve resiliency in older adults. And that's sort of where we're going with that. But I think, it gives us something that we could really work with to see if we can improve the response, whether it be to flu or COVID or any other kind of infection. So, and okay.
Thank you so much, Laura. I have two questions.
Mm-hmm.
One, I was talking to Luigi on chat during our sessions yesterday, saying to him, I think viral infections in older adults are a stress that is a measure of resilience. That measure of resilience he wants to look at. Because not only does the infection cause problems, but the older organism, whether it's an adult or a mouse, is less able to recover or it starts a spiral of health problems that aren't even related to the virus, but are because there's less resilience in the whole system.
Oh, I totally agree. Yes.
Right. So if you can, that virus is a stressor of the whole system.
Mm-hmm.
But then when I see your muscle effects, my question is that muscle effect this just readout of resilience in general that the older mouse can't cope with the stress of the infection, or is it a direct link to the T cell dysfunction and viral infection?
That's an excellent question. And I don't know if we know that just yet, but we do know that the skeletal muscle, the aged skeletal muscle responds differently to what's going on during infection than the young does. And we're just actually getting ready to submit that paper. We see different cells going into the muscle. We see different chemokines being produced in younger aged muscle. And what is driving that, we don't know. We know that there's no flu virus in the skeletal muscle. It's some systemic factors are involved here. And we don't have any idea right now what those might be and what is triggering the different response in the young muscle versus the aged muscle. But again, I think that this is one of the big things. As any stressor, and it doesn't have to be a flu infection, but any stressor that's going to induce systemic inflammation is going to have this impact and lead to physical impairments. And I think that's sort of where you start that downward spiral.
Right.
Yeah.
Yeah, totally agree. And my other question was, you looked a lot at CD4, and as you of course know, CD8s are also important for antiviral immunity. Mm-hmm. Have you looked at what happens to CD8 function in the older mice?
We have a little bit. we haven't focused on it because honestly, we were working with the vaccination, and since we're using protein vaccination, we were focusing on CD4s, but we do have a study that we're just wrapping up a little bit on the primary and memory responses of CD8s and how this is impacted by aging and senescence. So,
Great.
yes.
Terrific.
So, yeah, it's an amazing work. Congratulations, first of all.
Thank you.
And what I really start to understand, connecting it to autoimmunity also, is I think you see that there is a transdifferentiation of T effector cells towards Tregs and then vice versa.
Mhm.
So I think you're actually, what it seems to happening is that the T effector cells become exhausted. So I think that would be actually something that should be further investigated.
Mhm.
And I don't know what your ideas could be, because I think it's changing the phenotype of CD4 T cells.
Yes,
What I'm seeing
I
now.
totally agree. And my whole hypothesis is that the senescent environment that's happening in the aged lung, where the T cells are CD4s are differentiating, is a different cytokine milieu than what is going on in the young lung, and especially because of the SASP, and that's influencing T cells to differentiate down a pathway that they wouldn't normally go in a young lung where you don't have as much SASP. And
Exactly.
I think that if we can use different ways to target the SASP, whether it be with genetically modified mice or with senolytics, or if you want to block specific cytokines like we did with the TGF-beta, I think that this can turn around and sort of make the differentiation pattern more youthful.
Exactly. So I think it's something in the milieu that actually changes the phenotype of CD4 T cells, and that's why they cannot respond anymore.
Right.
So would you actually think of like CAR T cell therapy or something like that in these patients to make the immune response stronger?
But we know, and then I didn't talk about it, but we have published. So if you put young T cells into old mice, I did talk. If you put young T cells into old mice, they also don't differentiate really well.
So there are factors that they're suppressing.
Yeah. So I mean, it helps. It's a little bit better, but it's not.
Yeah.
And it's again, I think targeting just we're using sort of the T cells as a readout. I think that having targeting senescence and the overall effects of senescence might be a better approach.
Yeah, of course.
Yeah,
for sure. What about transferring B cells? Have you checked that? Are they also defective?
They are a bit defective. Technically it's not really B cells don't like to be transferred. There's a lot of death when you try and do that. And when I've talked to my B cell colleagues, they're not really thrilled about that idea. So I think, if again, other ways to address this, which some of our other panel members might have ideas about.
Yeah, of course.
would be a better approach.
Great. Okay, I think we'll move next to Paul Robbins, who has done some amazing work with senolytics, and he's going to talk to us about a Nature paper from his group that showed that just aging the immune system causes aging of multiple organ systems. That I think is an amazing paper. An amazing paper.
Great. Thanks, Joan and Georgina. This is a wonderful session you put together. So can you see my slides? Oops.
Yes.
Yeah. So I'll have to touch on the story that Joan just mentioned in the second related story. But where we started was in conjunction with Laura Niederhofer's lab, was trying to identify what senescent cell types actually drive aging and disease. And the way we did this was to increase DNA damage in specific cell types by the enzyme ERCC1 in the complex with XPF that's important for nucleotide excision repair, double-strand break repair, as well as interstrand cross-link repair. So you can cross this with a Cree driver and get deletion of a key DNA repair complex that leads to more damage, and in many cases, more senescence. So we've done this in a number of different models, the complex in a variety of cells in the kidney, in islets, in the brain, in the eye, etc. But the one that really gave the most impressive phenotype is actually when we deleted the DNA repair complex in all immune cells using the VAV promoter to drive expression of Cree. What we showed was that if you look in these mice, we call them VAV-I-Cree, ERCC1 minus flox mice, early in life, and these are just looking at CD3 cells, but this is true for other immune subtypes, there's really no evidence of senescence. But by 8 months, as you see in the red, we now see an increase in P16 and P21 in SAS factors, CD3 cells. But importantly, we also see the same increase in senescence in CD3 cells in wild-type mice. This is true with natural aging, as well as this increase in DNA damage to the immune system. But what was really intriguing is when we look in different tissues, we could see that in these mice that had a deleted DNA repair complex in immune cells, there was an increase in senescence markers of P16 and P21 in a variety of tissues, such as aorta, lung, liver, and pancreas. Showed this was not due to infiltrating immune cells, but actually due to non-immune cells within these tissues. And we also demonstrated that you could transfer either these cells from the accelerated senescence in the immune system or from old wild type into young mice, and these cells drive senescence from both models. And we see an increase in senescence again in a variety of tissues, kidney, spleen, etc. And we see markers such as P16, P21, and other markers of senescence go up. Certainly, if you actually transplant young immune cells in the old mice, we see a reduction in senescence. Senescent cells are being cleared by transplanting functional immune cells into an old host. So immune aging actually has two effects. It drives senescence in non-lymphoid tissues, and it reduces the ability of the immune system to clear existing senescent cells. So it's really two types of effects in aging. And what we observed is this increase in senescence, first in the immune system and then in non-lymphoid tissues, leads to a number of disease-related conditions, such as increase in liver dysfunction, increase in kidney dysfunction. We see loss of intervertebral disc. So this is disc aging. If we inject cardiotoxin in the muscle, that damage tissue. And importantly, these mice die at a much earlier age, and they do not die. It just seems to be accelerated aging that leads to their death. But importantly, based on studies that Joan has published a few years, we were interested to see if we could correct this defect in these mice that have increased DNA damage and increased senescence in their immune. And we did that by treating them for 6 weeks with rapamycin at a low dose, similar to what Joan has done both preclinically and clinically. And then we actually immunize the animals to an antigen, and then we inject the antigen to the foot pad. So this is the late type hypersensitivity model. And we could show that rapamycin treatment increased foot pad swelling in these and increased antibody production against KLH. So both a T cell mediated effect and a B cell mediated effect. And rapamycin had a set of different populations. These are actually PBMCs, and we could show there's reduction in these mice with rapamycin in P21. So this phenotype can be corrected in part using, in this case, a xenomorphic, but we have data now that also have similar effects to improve immune function. So I'd just like to close by discussing the second story. It's about aging and its effect on resilience, which was discussed as this session started. And this project was endemic. It was based on results we had obtained in a simple model system where we take an SPF mouse and co-house it with a pet store mouse that has a number of different pathogens. And surprisingly, at least to us, we find that young mice are fine. They have changes in their immune function, but there's no mortality. Old mice, 2-year-old mice die within 2 weeks after exposure to pet store pathogens. And what's intriguing is the major pathogen that's transferred between the pet store mouse and the SPF mice is actually a mouse beta coronavirus. This is mouse hepatitis virus. And we've shown that if you immunize them, we can decrease mortality in old mice, suggesting that it really was MHV which was driving mortality. And what we showed was, a week after infection or with exposure to the pet store mouse, there's a huge increase in old mice in particular. So this is shown in the black bars. And we can see this increase in senescence markers P21 and P16 in a variety of different tissues. So there seems to be a hyperactivation of senescence, and there's a hyperactivation of CC within a week after exposure to pathogens. So there's superactivation in old mice of senescence and the secretory phenotype. And importantly, if we show that if we cleared senescent cells from these old mice prior to exposure to these pathogens, in this case, we've used a genetic model where we take the Incattac mice with a drug that will specifically kill P16 positive senescent cells, or two different treatments with either the senolytic fisetin or the fisetin plus quercetin. We see we can protect about 50% of the mice from death. Why it's 50 freaking, and we don't yet have an answer for that, but this suggests that clearing senescent cells prior to infection or even after infection to improve survival and reduce pathology. So the model, and this is from the science, in young healthy individuals, when you're infected with a virus, the immune system functions as you would expect to clear the infected cell. Senescent cell burden, these senescent cells are hyperactive to PAMPs that are released by the infection. And this can lead to SAS factors, which interfere with immune function, which reduced antibodies and reduced T cell responses, lead to failure to clear the virus and increase mortality. And I should point out, we think there's senescent cells which are immune in senescence. They also secrete SAS factors that affect immune function. But if you clear these senescent cells, it reduces immune function and clears the virus. And importantly, these studies have led to 3 clinical trials to use the senolytic fisetin in different COVID patients. And these studies are ongoing. One of the clinical trials is close to completing enrollment. So we'll be able to break the code and know if there's a very shortly. And I'd just like to close by saying that even in young mice, we see there's an increased senescent cell burden to the pathogens. So this raises the question whether pathogen exposure increases the senescent cell burden, which then actually contributes to driving pathologies associated with long haulers. And there's a clinical trial starting or proposed to start to see if clearing reduces some of the long hauler syndromes. So with that, I'll close, and Joan and Georgina, if you have any questions.
No, just amazing work, Paul. I mean, for me, one of the most exciting papers that have come out in the aging space in the last few years is that paper of Paul's, again, showing aging of just the immune system triggers aging of all sorts of different organ systems. And you can see a decrease in function of the aging organ system. Just profound implications, because it suggests that the immune system is kind of a fundamental nidus causing aging, organismal aging, and something we, again, need to target therapeutically.
And obviously, immune cells can infiltrate into different tissues. So if they're senescent, expressing SAS, they're throughout all these tissues. So they're very in spreading additional senescence and driving aging pathology. Unlike a senescent muscle, etc., immune cells are everywhere. And we're now trying to determine what key senescent immune subtypes are important. So those studies are currently underway to see if it's all immune cell types, or if there's a specific cell type like a Treg or a CD4 or a B cell or an NK cell or NKT cell.
Great. So I think maybe, Georgina, what we'll do is let everyone go through their presentations, and then we can ask questions and have the panel. We want it sort of a group discussion.
So we should go to the next one?
I think so.
Sure. The next one is going to be Tim Peterson. He's a professor at the Department of Medicine in Washington University. So, Tim.
So Georgina and Joan and the rest of Metabesity, thank you so much for having me. I'm excited to be part of this panel. And I'll kind of just jump right into it with this slide here. So I'm really interested in drug mechanisms. So this talk is really going to ultimately conclude on drugs and hopefully some interesting ways to identify senolytics. But we start everything we do with the genetic approaches. And in this case, what I'm showing you on this slide here is a bunch of screens done by a bunch of other colleagues of mine. We do a lot of these genome-wide screens with CRISPR these days. And we do ours mainly with small molecule drugs, but I was very fortunate to work alongside Tyne Brumelkamp and Jan Corrette back at MIT back in the day. And they had really pioneered a lot of the genome-wide approaches that many of us all use today. And many of these papers, many in the top journals here, the PubMed IDs are listed alongside them, helped identify some of the key host factors, which are important for a variety of pathogens, viruses, bacterial toxins, et cetera. So, at the start of the pandemic, a lot of us kind of were thinking about, how can we contribute to the study of infectious disease. I really didn't have much experience with it beforehand, but I kind of asked the very simple question, okay, so we take all these papers, all these really great papers over the last decade or so, and can you all see my cursor here? Might hover over the right side of the screen. Okay, great. So we just asked the simple question, if you take all these different pathogens, what are the common human or mammalian genes that come up when you screen against those pathogens? So that was the basic question. So we had been doing, like I said, we've been doing a bunch of screens with small molecule drugs. And just to jump right to the point, just so we can focus more on the discussion. So we've been doing a bunch of screens with these drugs that we call CAD drugs, cationic amphiphilic drugs. And I'll explain more in the next slide what those are. But essentially, when we screened, we've done dozens of screens, and we looked at other drug screens other people had done. Essentially, the top hits from the screens with the pathogens were the same as those that we identify with this the CAD drugs, the ones that have this cationic amphiphilic chemistry. So it's a very specific response. The top hits, is 80% overlap of the top hits. Very exquisitely sensitive, specific results. So that really got us excited there. So, okay, so what are these CAD drugs? All right, so there are a lot of things that we know about. Actually, a lot of the most well-known senolytics, dasatinib, navitoclax, are CAD drugs. So the CAD drug basically has two properties. They have a high C log P, so essentially it's like they're more hydrophobic. So they partition an octanol relative to water relatively a lot. So a value of 2 is kind of the coefficient there. That above 2 is what we're looking for. And they have a high pKa, so they're essentially they're basic. And in the acidic organelles, they get protonated. Those are acidic, and so they get trapped in those organelles and disrupt the biology that happens there. So there's a lot of other kind of famous drugs there: sertraline, fluoxetine, which is Prozac, hydroxychloroquine, kind of a infamous drug from the COVID days, amiodarone, which is a well-known drug for in cardiovascular space, verapamil, blood pressure, a variety of blood pressure medications. And then a lot of the other famous drugs in the longevity space, metformin, rapamycin, of course, not shown here, alendronate, one of the most famous bisphosphonates for osteoporosis, and one of the, all the statins are not CAD drugs. So, and so in terms of the chemistry, like I said, they're basic, and so they always invariably have this have an amine that gets protonated, and they have often they have some phenyl rings, which are kind of confer the kind of the greasy hydrophobic part that allows it to, in many cases, pass through the blood-brain barrier in the case of the psychiatric drugs. Okay, so we not only did, so that was from unbiased genome-wide screening. So again, we weren't looking for anything, but essentially we kind of found this strong overlap of a chemistry in specific drugs with a wide variety of pathogens. So then the next kind of simple thing we did here was just to, again, do a similarly kind of unbiased transcriptional profiling of CAD drugs and pathogens, taking all the CADs that we had done transcriptomics on, and similarly taken all the pathogens that we had profiled in using genome-wide CRISPR screening, and but instead this time just use transcriptomics. And essentially those signatures were also highly overlapping. Maybe not surprising given the CRISPR screen results, but nice to see that at the transcriptional level, there's a high concordance between the gene sets. So a lot of our famous friends, the SERPINE PA1, P21, and others are in this set. And then in terms of the molecular biology.
I'm sorry to interrupt. Are you changing your slides or?
So essentially, this is just a heat map of the. I mentioned that we had done. Other people had done CRISPR screens with pathogens. We had done screens with a variety of drugs, and essentially there was a very high level of overlap, specifically with the CAD drugs, the ones with the cation epiphilic chemistry. These gene sets. I'll explain more in a second what they are. These are the CAD drug chemical structures. They may all look similar, but just trust me that the CAD drugs have this high C log P greater than 2 and a high pKa greater than 6.5 or so, give or take. So that's the unique kind of physiochemical properties of these drugs, which have this overlap with the pathogens. And then the same result at the level of the transcriptional profiling, very similar gene sets, highly senescence related, and then like I just alluded to a few seconds ago, there's a strong overlap with sphingolipid biology. And so sphingolipids are essentially cation epiphiles themselves. So there's really what we often see in these screens is the endogenous biology is basically competitively inhibited by drugs we throw at these cells. And so in that case, that made sense. And sphingolipids are, of course, are highly influential in the senescence response and in the lysosomal biology where these drugs are active. So that it all checked out. But I think was really amazing about both the genome-wide screens and the transcriptomics is just the degree of similarity. Like you just can't. There's kind of no debating, what the gene signature was in both cases. And so just in summary, I think probably this first bullet point is if you kind of learn anything from this talk, I think from, being a relative newcomer to the field, looking around to see, okay, there's all these senolytics, but, there's just kind of no logic to identifying them. And we can do screens, where we're measuring the proliferating cells versus the senescent cells and trying to selectively kill the senescent cells. But essentially what our results say is you actually don't need to do an experiment at all. You just simply need to look at the pKa and the C log P, and just those two chemical properties will predict the senolytic activity. And we've gone on at my company BioIO to essentially prove that. And so we'll have work forthcoming on that. And so this works not only just for pathogens, but every other senescence inducer we've checked. So we think it's we've identified a kind of broadly interesting class of senolytic drugs. And then maybe one validation to kind of tie it back with pathogens. This is work done by a colleague of mine, Eric Lentz, who kind of upon our work building at least the molecular basis building, he initiated a clinical trial with the CAD drug fluvoxamine, which is an antidepressant. He's a psychiatrist, and to my knowledge, and this drug that they were able to produce a really nice JAMA paper 2 years ago, I think now, where fluvoxamine was actually effective in the context of COVID, and several other groups have subsequently also validated that result. And to my knowledge, it's really one of the first and only repurposed drugs to have shown promise for COVID. So that was pretty exciting to see that the molecular details play out in the clinic. And just if anyone wants to get in touch, talk later, I'm generally available at Tim R. Peterson across the internet. So thanks, appreciate it.
Tim, so cool. I mean, seeing all these drugs that you would never predict, like fluvoxamine, probably going to be not only anti-COVID drug, but in senolytic. It's amazing. Your sort of new approach to drug screening. Just quick question. Did you mention rapamycin that is or isn't a CAD?
That's not a cat drug. Yeah.
Oh,
That's
too bad.
yeah. Well, it's good. I mean, it's good. You don't want everything. So actually, what's interesting about that is, so I mean, I've come from the studying the TOR pathway, and we haven't really investigated the TORC2 pathway, but essentially, if you look in the yeast literature and in other places, that sphingolipids are highly tied to the TORC2 pathway, the rapamycin insensitive pathway. So I think in some sense, all these CAD drugs are essentially could be thought of essentially as kind of, I don't want to say selective TORC2 inhibitors, but again, I think the sphingolipids biology is very tied to the TORC2 pathway. So it's kind of an interesting approach for targeting TORC2 when we're really, there's not really an approach for that currently. So
fascinating. And then when we get to the discussion, I'd love to hear all of Paul's senolytics, whether they fit the high C-log-P, high pKa. Yeah. So interesting.
A lot of them, like I said, Navitoclax, the dasatinib, most of the kind of popular ones do. So it gives a logic to why those actually are successful. So, yeah.
But what's also interesting is the fact that the connection that they see with antidepressants.
Oh, yeah.
So it's how the nervous system affects the immune system. So this is
Definitely.
really very, 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 very interesting. And I think this has a lot of links with David's work on.
Yeah. And then if you ask any, the neuroinflammation field was not popular.
Yeah.
The monoamine hypothesis for depression and a lot of psychiatric disease was kind of the dominant thing. But now the kind of neuroinflammation, so all these is gain interest. So I think psychiatrists are very open to this idea that inflammation and senescence factors are key to depression, and of course also depression is more common in older adults too. So it all kind of checks out.
And also for protective immunity, that would be interesting for Laura's and Paul's studies as well.
Yeah.
Okay. So.
Thanks.
Sure. So our next speaker is David Furman, who's director of the Stanford 1000 Immunomes project. And David is sort of a pioneer in using AI and big data in analyzing immune function and its decline with age. So David, all yours.
Yeah, I should also say that aside from my Stanford hat, 3 years ago, I was recruited by the Buck Institute for Research on Aging. And I'm leading an AI platform there to better understand aging. And I'm also associate professor at the same institute. So what I'd like to bring to this meeting today is the idea that non-communicable diseases and infectious diseases share a common denominator when you look at aging, and that's systemic chronic inflammation. All of you know about systemic chronic inflammation and the effects on aging, or most of you in the audience. But I'd like to stress that it really connects both non-communicable and infectious disease. And I have some data to share today. You have probably all seen this graph that was published in 2013 from a Cell paper on the 9 hallmarks of aging. The word inflammation was never even mentioned in the whole paper. To my surprise, it was shocking because systemic chronic inflammation is actually triggering each one of these hallmarks of aging. And there's quite a few papers demonstrating that it leads to genomic instability, altered communication in between cells, exhaustion of stem cells, cellular senescence. It goes together. We have data showing that endothelial aging drives cellular senescence in a paracrine fashion, and then mitochondrial dysfunction, etc. And so I think now most of the folks in the aging space have acknowledged that systemic chronic inflammation is actually leading the way. So back in 2007, when I arrived at the Stanford community with Professor Mark Davis, we decided to reinvent the way we do immunology, really, and focus in the immune system of humans. We've heard great things from Laura and from, sorry, Tim, etc. But mice are a little bit different from humans, and in different diseases, things can or cannot translate. And so we know that for the most part, immunological models of mice are not ideal. And so we decided to really go and focus in humans and do multiomics. At that point in time, we were using proteomics and metabolomics and transcriptomics, etc. But the technologies really evolved. Proteomics was a 45-plex Luminex kit. Now we're measuring 8,000 proteins. So things changed, but the premise is exactly the same. By analyzing multiple probably hundreds of thousands of parameters from large cohorts, we were able to understand the immune system a little bit better and translate into diseases of aging, in particular aging of the immune system at a higher success rate. So the Stanford 1000 Immunomes was born there. We secured more than 45 million from NIH and other institutions, and we're now continuing the study for the next 5 years with money from NIA and NIAID, which both funded Buck Institute, Stanford, and Technion in Haifa with a collaborator, Shai Shinoa there. So we've been looking at, again, transcriptomics, proteomics, all the multiomics since the very early days of human immunology. One of the things that came as being extremely significant of all the work we've done over the years was a new clock of aging that's based on proteomics. So we call that inflammatory age. And I'm going to refer to inflammatory age as IH from now on. So based on the 1000 Immunomes study that I'm showing in the far left, top left, we used a deep neural network type of approach. It's called guided autoencoder. And we tuned that so that the protein data that was found in the blood was predictive of the age of an individual. And at the same time, the computational task would ask the question, can you recapitulate the original data? So we're trying to have the immune age index by measuring cytokines and trying to predict age, and at the same time reconstituting or reconstructing the cytokine data. And so we're using that type of a model to build what we call the inflammatory age, or IH. And that's shown in the far right on top, the IH metric in the y-axis and the calendar age in the x-axis. So we can predict age, but that's nothing really difficult from a computational standpoint. Using blood biomarkers, anyone can predict age. The important part is, once you predict age, is it the variability in the inflammatory age, in this y-axis, that variance, is it explanatory of diseases? And that's what we did. That's shown in the bottom left. We can predict multimorbidity, very important in the aging space. We can also associate with frailty. That's right in the middle. There's a typo. There's a problem there. It says multimorbidity, but it should say frailty. So what we have done for the frailty analysis is looking at predicted frailty score in 2010 in the y-axis and the observed frailty score measured in individuals, in actual people over 70 years of age. And we can predict frailty about 7 years before it happens. And we can also associate that with centenarians. And by building a gene expression surrogate of inflammatory age, we also predict all-cause mortality in the Framingham heart study. So what I'd like to stress again, as I said at the very beginning, is that systemic chronic inflammation not only has a bystander effect in tissues and organs and deteriorates different physiological systems, that in the aging space, but also it also deteriorates the immune response. So there's been quite a few papers showing that baseline inflammation predicts poor responses to vaccination and infection. And we actually published a paper with Shai Shinor back in 2016, I think it was, or 15, where we proposed that chronic inflammation saturates the immune resources. In particular, we looked at the phosphorylation of STAT proteins in T cells and B cells and monocytes and so on. And what we see is that cells from older individuals that are not doing well in their cardiovascular system, as well as in the responses to flu, have a hyperphosphorylated status in T cells at baseline, and they don't respond to cytokine or other stimuli after you take these cells and put them in culture. So we have this model where systemic chronic inflammation leads to constitutive activation, and then what the cell will sense ultimately is that fold change, not necessarily the absolute phosphorylated status of T cells, etc. So when we look at inflammatory age and we stimulate cells from individuals, and this is done in 1,000 people. So this is a single plot that took us probably a couple of years to run in terms of the experiments there. And we stimulated CD4, CD8, the B cells, et cetera, with different cytokines and looked at the phosphorylated status of signaling proteins intracellularly. And what you see in this plot with bars to the left and to the right is the regression of inflammatory age against all these different stimulations. So the higher the inflammatory age, the lower the immune responses to multiple stimuli in multiple JAK-STAT pathway, JAK-STAT, STAT 1, 3, 5, etc., and in multiple cell types, as you can see here. So overall, high baseline inflammation leads to poor responses to viral stimuli, and also very likely because the JAK-STAT pathway takes care of immune surveillance against cancer, susceptibility to cancer. And so what you see on the left-hand right of the slide is just simply an association between inflammatory age and the naive CD8 T cell frequency in the blood. So it's also a predictor of lower naive T cell frequency. I just wanted to jump very quickly before I open the discussion here with everybody. That we're now using spaceflight to understand accelerated aging. A year and a half ago, NASA reached out to me because they saw that astronauts are suffering from inflammation and aging in an accelerated fashion. That's actually what happens here. So what you see in the right-hand side of the slide is a plot where the different systems of the body are plotted against time in years, normal aging in the dashed lines, the gray lines, and accelerated aging during spaceflight. So it also accelerates metabolic rate, bone density, all those things decline. And when you look at the immune system, that's also true. So you have reactivation of herpes viruses and also inflammaging. So the exact same thing that we see in humans during the life course, you see it at a very, very accelerated fashion, about 30-fold during spaceflight. So we've been working with my collaborator, Chris Mason at Cornell, to analyze all the Inspiration 4 data. We also have the analysis of the twin data. You probably know the twin study data on NASA. We apply the SOMA, which is standard omics measure for astronauts. And you can see all the different time points before, during, and post-flight. And we're collecting all sorts of different biospecimens, and we're doing multi-omics on this type of specimens. Just to wrap up, I just wanted to mention that we can also accelerate this in a dish. So we are using human organoids and PBMC and subjecting them to microgravity in vitro. And what we see here in RAMP, that's our new model. I started a company based on this thing as well. What you see is that we can accelerate. So coronavirus pathogenesis pathway is highlighted here in orange. That means that it accelerates, that it's elevated with this treatment of microgravity. And we have treatments that we've been using based on machine learning algorithms that can completely abolish or reverse these different pathways. And that's part of the what we're doing here. The other last thing to wrap up is the new clock that we have built based on NASA Gene Lab. This is used in animals. So this is transcriptomic age in the y-axis and chronological age in the x-axis. And the time in space in mice that were flown in space, you can see that it accelerates very rapidly and then it decelerates. So it depends on the time in space, the acceleration of aging. And overall, when you take those genes that define that age acceleration, the most significant part of the physiology that gets dysregulated is the immune system. That you see in blue here, there's a decline in the immune response, and normal aging that's also happens with the space aging that gets accelerated as seen by this difference between the two in orange color. So with that, I'd like to just wrap up on that presentation, and I'll stop sharing screen.
That's fascinating. David, a couple questions, and then I want to open up to the panel. So for the changes that you see with the IA clock, how do we know which of those are compensatory and which are deleterious, or you don't know?
Are you referring to the actual analytes or the pathways?
Yeah, like when you look at the analytes, I'm assuming some of them are protective, upregulated to sort of compensate for frailty, and some of them are deleterious. Can you sort of sort it out? And are there any ones you can tell us about? What are those analytes?
Yeah. So the inflammatory age clock has multiple cytokines and chemokines and growth factors that were measured there. Some of them go down with age, some of them increase with age. So this idea of inflammaging, that everything goes up, is actually not true. So say things like CXCL9, it's a protein that gets activated in response to interferon. However, we're discovering interferon-independent mechanism when we age the endothelial cells that are the large producers of CXCL9 ex vivo. We don't see any gamma interferon in our culture. So the model is a little bit different. This is not infectious. It's driven by sterile cues. So that's really interesting. And there are other cytokines that decrease with age. The only one that we could effectively validate just because of funding and time was CXCL9 as one of the major drivers of endothelial senescence and arterial stiffness and various poor functions of aging, including cardiac hypertrophy. So that's one cytokine you don't want to have in higher levels. Eotaxin 1, it's also one cytokine that increases with age and has been shown to participate in neurodegenerative diseases. And we have another paper now on PNAS that's almost published there. It's been accepted, where we have a new clock that predicts brain aging. And we've seen eotaxin coming up again and again.
So David, one question that I really I wanted to stress, when you go back to your slides, I think that you have something involved with innate memory or innate tolerance. So what I'm seeing here is that you have some of the myeloid compartment that it's tolerating, and maybe that's because of the senolytic of the stuff that is being secreted by senescent cells. So you may have some sort of tolerance from the innate immunity.
Right. So if I can pull up that plot, but basically what happens is most of the adaptive immune response is down with high inflammatory age, but the innate immune response is actually it's the opposite direction.
Exactly, that's what I think.
Yeah, so monocytes and NK cells are actually ready to go. If you just touch them with a single cytokine.
Memory, yeah, exactly.
Yeah, they have much more activated than in people with low inflammatory age. And I think what happens is, in a way that tells me about susceptibility to cytokine storm. Because cytokine storms, when you have monocytes, macrophages in the lung, they produce huge levels of IL-1 beta, TNF alpha, IL-6. And so these cells, when you take them from the blood, they're ready to go in people with high inflammatory age. So in my view, it would be really interesting to see if inflammatory age is predictive also of COVID complications, because my feeling is that that would be the case, because you have a lower immunity to viral infections, but at the same time, a higher possibility to produce cytokine storm after the cells are activated there.
And do you see any phosphoproteins increase besides JAK-STAT? Any other pathways are up?
We haven't looked. We haven't looked. The most recent panel that we have, it includes mass cytometry panel with many different intracellular proteins that get phosphorylated, but haven't looked in detail for this project because we wanted to be consistent across years. And so we only had data for STAT 1, 3, and 5. The newer data uses less of it. We have less sample size, a very small sample size. So,
great. So,
one last question. It's more to Paul, but also maybe David can answer it as well. Because I'm new in the field of immune aging, I'm more of an immunologist. Are these substances secreted by senescent cells are they acting as dumps or pumps? What would you think? What is actually signaling on immune cells?
It's a great question. I mean, we use LPS as our model PAMP, but obviously there are many others out there. I guess HMGB1 is one that Judy Campisi has characterized extensively. It's a SAS factor and can activate senescence and distant cells or neighboring cells. So, and then Jim Kirkland has shown actually that the spike protein of SARS-CoV-2 functions as a PAMP to activate super activation of senescent cells through a mechanism that's still to be worked out, but probably involves a TLR, probably TLR3 or TLR4.
So now we want to open it up to all of you experts. One of the things about Metabesity is to see if we can have sort of a group think, sort of cross-fertilization. I mean, and I guess some sort of common themes I'm seeing are lack of resilience of the organism due to chronic inflammation causing not just increased susceptibility to infectious disease, but all sorts of other aging-related diseases. How do we target this? What should we be targeting if we want to improve the function of the aging immune system? Should it be the CD4s and their inability to respond to vaccination? Should it be the senescent cells? Should it be other cells that are producing chronic inflammation? So, Paul, Tim.
I'll let Tim go first because it's a tough question to answer. So go ahead, Tim.
I'll take the easy answer, all the above, right? I mean,
yes.
I think I always reference the Gompertz law, essentially, right? The mortality is reaches 100% by the time we're all 100 years old. So essentially, and that's an exponential curve. Exponents involve multiplication. multiplication, implies synergy. I think we're going to need multiple synergistic factors all working together to make a substantial effect on lifespan.
And Paul?
So all the above. Yeah.
The big question that the field has is what type of senescent cell is the key target? Is it any senescent cell? If you have senescent on the brain or fat or muscle or liver, does that drive immune dysfunction, or do you need senescent immune cells in addition? And the field doesn't know that. And we also don't know if you use D plus Q or fisetin or CADs, whatever, what senescent cells are being targeted in vivo. So we see improvements, but I don't think we really know what the key target is. And then, we're not developing drugs that target those key senescent cell types. I think one of the things that's happened in the field is that the NIH Common Fund has put 180 million into identifying and mapping spatially senescent cells in humans and in mice. So I think we'll start to learn much more about what type of senescent immune cells we can detect in different tissues, as well as non-immune cells. And hopefully that information will help us lead to better drug development that can target the key cell types. But right now, we treat with D plus Q and it's a systemic effect because we don't know what cells are being targeted that then improve immune function.
And is all chronic inflammaging due to senescence, or is some of it just due to hyperactive monocytes or macrophages or dendritic cells or immune cells spewing out too much inflammatory cytokines?
I was asked David that question myself because it's a chicken or the egg. I view all kind of inflammaging due to an increase in senescence, but we know inflammation increases senescence, so it's the chicken and the egg. And so I don't know what starts that process.
Yeah, I'd like to comment on two aspects here. One is the problem of the chicken and the egg that Paul is mentioning. We know that senescent cells, wherever they start, it could be fibroblasts, could be endothelial, they become senescent and they become inflammatory. So in our hands, the inflammatory aging metric shares about 45 to 50% of the SASP. There are things that are independent, there are things that are shared, right? Okay, so and that leads to more senescent cells, at least in cultures, and we believe in vivo as well. That has yet to be defined, as Paul alluded to. Now, what to do? Where does this whole thing starts? I think there's been, the ideal world would be to identify those cells that are producing these nasty pyrogens and try to eradicate them. But that's not going to happen anytime soon. We don't even understand where they are located, which cells are those particular cells that are producing most of these chemicals. So one thing that Laura has very beautifully started a while ago, 15 years ago or more, was to take cells in general and deplete the whole thing and then let the mice, in this case, repopulate. And that in itself, it's a huge achievement in the field of immunoaging, because she was actually able to reconstitute a very healthy immune system in those mice. I think it was a JM or JI paper. It's pretty old now, 2004, 2005. But then also the work from Irina and Michael Comboy, where diluting plasma, everything is in the plasma, just diluting them enables rejuvenation of the immune system and other physiological systems as well. It may not be the final answer, because if you have those cells that are still producing these chemicals, then it's only transient. So you would need multiple rounds of dilution in your plasma, but at least it's something that we could do today. And I know there's a company called Lifespan that started to do this in humans, where they're basically diluting plasma, adding some goodies there. And all in all, you see major rejuvenation effects. But they have to come back, obviously, for the new round of plasmapheresis.
I don't think that's been placebo controlled either, correct?
Now it's being tested as we speak. So
Yeah,
there's
so I think we have to wait and figure out, have a
there is
control group.
group.
Yeah.
Exactly.
Yeah. So just to bring in Tim, okay, so we're hearing all about Inflammaging, but your CADs, as I understand it, are acting directly as antivirals and antimicrobials, or is it via the immune system?
What's the
And
mechanism?
how is senescence tied in?
Yeah, so I mean, essentially these drugs they're competitive inhibitors against sphingolipids. Sphingolipids are, one of the two major components of lipid rafts. Most pathogens enter through lipid rafts. They're really just disrupting the endosomal, pathway by which pathogens do their damage. And then that just also happens to be, there's not only used by pathogens, so this also used, by other forms of damage that that then lead to senescence of the cells. So there's kind of a confluence of things that are happening here in this mechanism. I realize it's kind of more complicated than maybe.
Sorry. Why are senescent cells so sensitive to CADs? What is it about the sphingolipid in the endosome and the senescent cell? And apparently normal non-senescent cells aren't sensitive to this.
Yeah, so I mean, in terms of which sphingolipids accumulate, we haven't gotten that far yet. Yeah, I mean, it's kind of that's definitely an open question, but Sarah Spiegel at VCU has kind of some seminal work on the role of sphingolipids in senescence and driving, I think, P21. She has a science paper from 2008 or so. So they're more, she and the sphingolipid biologists are much more of an expert on this stuff, but we're just kind of coming in with the correlations we've identified, and we think they're pretty strong. And yeah, like, again, I think what is exciting for me from entering this field is like, okay, I finally feel like there's like a logic, there's like a, molecular logic to designing these senolytic drugs, right? And so it's just this simple kind of two component pKa C log P, and then you're basically guaranteed to have a senolytic. So
You go. I'm
One last
laughing.
question. I mean, this is so fascinating. But then, Joan, we have several questions from the audience. So one last question to Paul, and then I'm going to shut up. I promise. So, Paul, what I really loved about your work with the specific pathogen-free mice and mixing them also with the pet store mice was probably that there would be an effect on the microbiota.
Right.
So I think you're changing something there too, apart from the virus.
So it's a great question, a great point. We have not looked, but having said that, we have published recently in collaboration with Milan Masternak that treating old mice with D+Q changes the microbiome. So actually reducing senescence affects the microbiome. So the corollary or the reverse would be more senescence is probably changing the microbiome, as is exposure to pathogens. We've not started to look at the microbiome. You can only do so much, and so we've not focused on that. But I agree completely.
No, I know. I
It's probably changing it dramatically. That's probably affecting senescence, inflammation, immune responses, etc.
In testing immune responses, for sure.
Yeah.
And one more question. Okay, there's very few senescent cells, right? We think in
Right.
humans, but there's a lot of inflammaging and elevations in cytokines. And there is David, you said, well, there's a 50% overlap in what you see in inflammation and what you see with SAS, but SAS can also be molecules produced by regular or old inflammatory cells in the body. So I'm still trying to figure out how much of inflammation is senescence and how much is, and maybe we just don't know, but I would assume some of it has to be beyond senescent cells because there's so much cytokine increase. Am I correct?
David, do you want to start? You want to handle that one first? I mean, obviously, macrophages can be P16 positive, but not necessarily senescent.
Yeah.
And as Dave was saying, that they're primed to respond. Are they, pre-senescent, whatever? That's something we're trying to define through this this consortium, the SENNET consortium, to identify and characterize and map senescent cells. So, I don't know, David, your thoughts?
Well, I think there's a lack of consensus in the definition of senescence. We all talk about cell senescence, but the concept of immune senescence is slightly different, and some people are referring to both interchangeably. The fact that the immune response declines with age, that's immunosenescence. That does not necessarily mean that we have senescent cells in the body. We think that that's the case, but cell senescence in the immune system has, I don't think has been well defined whatsoever. So what we know about cellular senescence has mostly come from in vitro systems with culturing, fibroblasts and mesenchymal cells and irradiating them. And then the whole work of cellular senescence has been built on that premise. But I don't think we've seen work on cellular senescence in the immune system that can define better which of the subsets that express these different. We don't even know what the markers are, quite honestly. We're using P16, P21, beta-gal, but those things are also expressed in other cells that are not senescent. And so we have to first define what's the minimum panel that will work for this particular cell type, which is in itself a big undertaking. And then start asking questions as in, what are the immune system cells that are senescent in an individual? So we're just starting it. That's my, I'm sorry, I can't be more specific there. Yeah, Laura, please help me here.
Yeah, and then the other issue that I grapple with a lot, especially looking at a viral infection in the lung, is that when you have injury, you're going to have senescent cells that are part of the healing process. And I know Judy Campisi talks a lot about that with using her models. And that's a transient senescence that's involved, and then it's cleared. And I think, having this transient senescence involved in healing versus sort of chronic senescence, for lack of a better term, that's involved with aging, may be two separate things, but also they may interact with each other, especially during something like a viral infection in an older individual. And that's going to be hard to tease apart.
And I think David's point, there's immunosenescence, which is a decline in function of the immune system, which might be very different than actual senescence of an immune cell with P16, P21, beta-gal. There may be some overlap and some not overlap.
Personally, I don't like to use the term immunosenescence because it does imply that cells are becoming senescent in the immune system, and I don't think we can actually say that right now. So, I always go with something like age-related changes, because that's a lot more vague, because really we don't understand what's happening.
Right.
Okay, so we're going to take some questions from the audience now. So I'm going to start with the beginning. I saw, David, that you answered a couple of questions. So one question is, are you developing any NCEs based on this data set? This is for David.
NCBs? I didn't
New
see.
chemical entity. New drugs.
New drugs from this data set.
I didn't see the question in the chat. We are, right, exactly. So the first work that I described on that inflammatory age, we have these cytokines that are largely contributing to decline in function, and we have ways to go from multiomics to interventions. And we are now in our second large clinical trial. That's part of my company, my first company, on the NASA research in the space biology approach. I have the second content we just incorporated, and that's exactly what we're doing. Drugs for ameliorating the risk for space flight and at the same time for accelerated aging as part of that effort. And so we have a number of candidates now. As I showed one of those that is reversing the signatures of aging, largely cellular senescence and also immune decline. But that's a first approach. That's first drug. We're now having multiple other approaches to identify drugs to target senescence and aging.
Okay, we have a question for the whole panel. Can you comment on the utility of targeting the NLRP3 pathway for inflammaging? Anyone want to take that on?
You know, that I at least can say that there's a lot of evidence now that the inflammasome plays a role in driving senescence, that there are links to the inflammasome in senescent cells, both in the primary and maybe the secondary senescent cells. There's evidence that the inflammasome plays roles in multiple diseases. So I think you would infer that targeting that would provide a benefit. Now, is there a detriment? You know, you always worry if you block some of these pathways, is your immune response going to be appropriate? And that's something I think you have to test empirically. So I think yes, the answer is most likely going to be targeting it would have a benefit at reducing senescence and improving immune function.
Okay, then we have a question. Any research on high fructose corn syrup, which has been shown to increase systemic inflammation with an increase in endogenous cortisol to cool down the inflammation? Any thoughts? And which I guess gets to nutrition and its contribution to systemic inflammation.
You know, obviously, some of the flavonoids that we're working on, their senolytics, are found in a variety of different fruits and vegetables. So, you know, obviously, there are things you can eat that might reduce your senescence, might reduce your inflammation. And there's correlated evidence that that's the case in people that eat a lot of strawberries, a lot of blueberries, that they have less P16 positive CD3 cells and less markers of inflammation. But when it comes to high fructose syrup, I'm not sure about that one. Yeah.
Maybe contributing.
And then another question.
Oh, go ahead.
Yeah.
Just real quick. I mean, the only factoid I know that can add to the conversation is, from what I know, you know, when you add sucrose and fructose to diets, you know, often that drives a fibrotic response. So I think if those of us in the space that are trying to target the fibrosis that happens alongside the inflammation, you know, sucrose and these you know, non-glucose sugars can be ways to drive that fibrotic response, and then therefore we could design drugs around that. So that's just a little factoid. Sorry.
Interesting.
Yep.
Okay, another question. Can you comment on the decrease of clonal diversity with age in hematopoiesis, and could SASP cells arise as a selfish selection?
So we are looking at clonal hematopoiesis in aged mice. And if you age them out 2.5 years, there's evidence that you can start to see signs of clonal hematopoiesis. We've not characterized senescence. You know, does that lead to a subpopulation of cells that are more prone to senescence or not? So we've not gotten to that point. You know, and I guess getting back to the question, you know, David was saying we don't really know what a senescent immune cell looks like. So we say senescent-like state. I think reviewers want us to put that in our paper. Was not senescence. It's kind of a senescence-like phenotype. And so we're just trying to characterize that in 2.5 and 3-year-old mice where we see more clonal hematopoiesis. And whether that is true also in humans, we'll have to see. But I think the answer is maybe.
I think Laura could also respond to that, because I don't think we see that much of them. Would you actually believe that, like, clonal diversity would switch cells toward more senescent immune cells? Or I would.
I'm not sure that there's any evidence for that right now.
Yeah.
You know, I think lots of groups are investigating it currently. I know Sapologist said he did. We're actually, I just actually got an email with a whole bunch of more data. And so I think it remains to be seen. It's a distinct possibility, but honestly, I don't know that anybody really understands how the decrease in clonal diversity is mechanistically working.
And how to measure senescence of hematopoietic cell progeny. You know, it's tricky.
True, yes, yes.
So last question. We have two more. So plasma dilution allowing for transient rejuvenation on a regular basis to reduce cell senescence volume might be a good thing. David, I think this gets to your comment that might be misinterpreted.
Yeah, I quickly responded to that in the chat box. I think we don't know yet. The clinical trials are underway. So the effects are very positive so far, but the clinical studies have not finished yet, and we need the placebo control that you just alluded to. So,
Great. Okay.
Okay, and a final question. Have you done a young to old heterochronic BMT, a bone marrow transfer to check lifespan aspects?
I don't work in mice.
Oh, no, I think this
No, I
is
think it's
for,
not for you, it's for everybody.
so in other words,
mean,
go ahead, Paul.
Yes, so we put young cells into young immune cells into a progeria model, and it seems to improve health span, but not necessarily lifespan. At least there wasn't a significant increase. So it appears to be a positive effect on health span if you put young immune cells, at least into a progeria model. We haven't done it naturally aged because you know how many times you have to transfer cells, et cetera. So we see a benefit on health span, not necessarily an effect on lifespan, at least in the studies that we've done.
You know, it's a really interesting question because when you age the immune system, you could see death occurring sooner, but apparently that decreased mortality can't be reversed with just young immune cells, and
Right.
or maybe it's two different models.
And it may be you have to get more infusions of young cells because you have old immune cells that may be affecting the function of the young. So their
Right.
activity goes down over time. So,
Right, and you know, you can get, if you put young stem cells into old mice, you can get highly functional CD4 T cells out, but it's not really a robust repopulation because of the old thymus. There's only so much an old thymus can do.
right.
And so, you know, with regarding T cells, but, you know, having it have younger innate immune cells and B cells might be really beneficial. So I think, you know, we like David said, we did these experiments. Oh, it's coming up on 20 years ago when we published them. So, and the technology is vastly improved. So it might be time to try some of these again.
Lord, so impressive to have people remember your paper from 20 years ago. That's like a really good paper. Anyway, we have to close this session. You guys were wonderful, so insightful. You know, we're so appreciative you came.